Okay, good morning, everyone. Welcome to the final lecture of CS 206. I don't know about you, but this is my last class for a while, so I wish you all a relatively gentle and successful end to your semester. Just a couple of announcements, obviously, at the end of the semester here. Just a reminder about what's expected from you to finish out this course. Most important thing is this coming Sunday, you are submitting two items to Blackboard. Your written report, a written documentation of what you've done in the final stages of your final project. And you're submitting a publicly viewable YouTube video. It is extremely important that you submit this at or before, or hopefully well before 11:59 p.m. On Sunday night. Amanda is the TA for this course. Amanda is going to have to stitch together 55 presentation videos between midnight and 7:30 a.m. So please be aware of what's required from you. At 7:30, I will put up a link to the oral presentation so you'll know exactly when you are unmuting yourself and talking over your 2.5 minute video. Reason I put up the presentation right at the end here is because I'd like all of us to be here for the entire oral presentation period, which runs from 7:30 to 10:15 in the morning. We've got 55 presentations to get through. We're going to stitch them together in a single playlist and play that playlist from beginning to end Monday morning. I'll put in two 10-minute breaks for us to break things up a little bit. And then we'll be all done. I will start grading immediately after the oral presentations finish, and we will try and get your grades up by Wednesday at the latest. Any questions about the final presentation? Everyone understand what's expected of them? If you're not clear on the details, there's the link to the final project and hopefully all the instructions. Everything you need is in there. All good? Okay, well, then, without further ado, final lecture of the course. Usually we would have had a 4-part section on evolving bodies and brains, expanding the evolutionary algorithm so that's able to automatically tune the body plan of the autonomous machine along with its neural network controller. Given the uniqueness of this semester, I've had to gray out a few of the lectures here. I forgot to put up the links for those of you that are curious about these lectures. You can have a look. I will put up the links after class today. Okay. Last time we looked at, is there no attendance sheet for today? Great catch, Sid. Thank you for reminding me. I forgot to make an attendance sheet for today. So give me a moment here and I will add one and then we can all signal our attendance. Give me one moment here. I knew I forgot something this morning. Okay, you can go and mark yourself as present. I'll give you a minute or two to do that, and then we'll jump back to lecture. Okay, All right, so in this last segment of the course, we're looking at our evolutionary algorithm that is tuning body plan and neural controller simultaneously. We looked at the first attempt to do this last time, which actually dates all the way back to the beginning the beginning of the field of evolutionary robotics in the mid 1990s, actually 1994. Definitely this experiment was way ahead of its time. There have been many projects since Carl Sims's work that have attempted to expand an evolutionary algorithm to modify body and brain. As you can imagine, it becomes a much more complicated evolutionary algorithm, but not that much more complicated. Some of you are tackling the bodybuilding final project, in which you do this in a modest manner. Today, we're going to talk about Xenobots, which is probably the most recent addition to the field of evolutionary robotics. This is some work that my group led along with a group at Tufts, which we'll talk about today, and probably exemplifies what makes evolutionary robotics and this sort of approach to AI and robotics in general unique from other approaches to robotics and machine learning and neural networks that you may have heard of. So I think these two lectures here are very good bookends for illustrating at the beginning and today what makes evolutionary robotics unique. Okay, so the nickname for these things are Xenobots. The more formal term is computer-designed organisms, because as we are going to see, the organism, which you see on the right here, was not designed by a human being. It was designed by a computer, where computer here is really short for an evolutionary algorithm. So these are really evolutionary algorithm designed organisms, but it doesn't fall as trippingly off the tongue as computer designed organisms. So we're going to go with CDOs. As I mentioned, this is work that was carried out by myself and my now postdoctoral researcher, Sam Kriegman. Sam took this course a few years back. I don't remember if he guest lectured for us this semester, but he definitely has in the past. 2 years ago in this class, he guest lectured and showed evolutionary robotics in which the robots were built not from metal and plastics and ceramics, but they were built up from biological cells. And I think this class was the very first one to be exposed to computer-designed organisms about 2 years ago. In summary, we're going to see an evolutionary algorithm applied to a physics engine, not the one you're using, but a different one that is able to evolve virtual robots in simulation. And then we were able in this project to successfully cross the simulation to reality gap, the sim to real gap. And our biology colleagues at Tufts, Doug Blackiston and Michael Levin, were able to build up our evolved blueprints cell by cell using frog cells. These are cells taken from the particular frog Xenopus laevis, thus the name Xenobots. Before I talk more about computer designed organisms, however, I want to just pause for a moment and show this plot. I usually show some figure like this at the end of every course I teach. Lots we can pull out of this figure. The main message here is some of you are trying to determine what you're going to do this summer, next fall, the year after, and so on. Some of you may go work in industry. Some of you may carry on in grad school. One of the main messages you can take away from this plot, actually the first one, is that the glass ceiling unfortunately still existed, was alive and well in 2012, and as we know, it's still with us today. Lots of interesting patterns you can pull out of this data. The most important one, I think, is that the gap between your economic health is much more probable if you carry on with grad school than even just a bachelor's degree. So for the seniors here, even if you're going out in industry, I hope you will consider returning to academia and carry on with a master's degree or possibly a PhD. So there are real economic reasons to go to grad school, but today we're going to focus on not the economic reasons for going to grad school, but the fun reasons for going to grad school is that you might get involved in a research project that other people find interesting, which ended up being the case in the Xenobots. This doesn't happen all the time. Doing research is intellectually rewarding in and of its own right, but it's obviously nice every once in a while if other people find what you're working on interesting. And that was definitely the case in the Xenobots, much more than at least I had expected. We published our first paper on the Xenobots back a little over a year ago in January 2020. Last month, we published the second paper, and we got some global media attention. I guess if you make millimeter-sized robots out of frog cells, you're bound to get some interest. I ended up on CNN last year. The New York Times covered our work, the BBC, and in this month's New Yorker, if you can get if you can get a handle on it. There's an article about Mike Levin, who is our senior biology colleague over here at Tufts. Mike's lab is doing some amazing things in biology, computational biology, synthetic biology. For most people, what they find most interesting about Mike's work is the promise that it might lead to the ability to regenerate lost limbs or organs, or regenerate brain tissue or other organs after damage or cancer. Some amazing things going on in his lab. So a few years back, my group teamed up with Mike's group, and we learned a little bit about what they are able to build out of cells in their lab, and we showed them a little bit about what we could evolve in simulation, and our two groups eventually met, and we eventually crossed the reality gap about 2 years ago and reported on this work. Okay, so that's what we're going to talk about today. So let's skip now into the technical details. There's a fair number of computational and biological parts to this project. So I'm going to focus, obviously, today mostly on the computational and evolutionary robotics side of this project. There is a lot of biology on the other side. I'm going to just sort of give you a taste for what is going on the biology side of this project. The reading for today. The reading for today is the paper itself. So there's lots of links into the biology side for the biology students here, those that are interested in biomedical engineering, synthetic biology, prosthetics, brain-computer interaction. You might want to spend a little bit more time with this paper. You might not have time this week given exams, but maybe over the summer. I'm happy to chat about this work and give you some more pointers into the biology side of this project and related projects. As I mentioned at the beginning of this course, evolutionary robotics is an extremely new field, and it's just in the past few years that evolutionary robotics has really begun to collaborate with biology and transfer some of the things that we do in simulation and robotics into biology. So for those of you that are thinking again about grad school or industry, there is some amazing things happening at the intersection of AI and biology. This is one of those intersections. Okay, so back to the computational side of things. I'm going to review. There's quite a few number of moving pieces here, but some of them are going to be very familiar to you. The first one is that we're going to use compositional pattern producing networks. Pattern producing networks, or CPPNs, to generate our virtual Xenobots. So just a refresher on what CPPNs are. They're networks. They're very much like neural networks. You could consider them neural networks if you like. They usually have an input layer and an output layer. In my little cartoon here, we have a 2-neuron input layer and a 1-neuron output layer. We define a 2-dimensional space in this case, and we visit a number of points that are regularly spaced inside this space. And at each point inside this space, we supply the coordinates of that point to the input layer, and the value or values that arrive at the output layer, we then use to paint something into that space. When I introduced CPPNs to you before, we were painting gray into a space, or the amount of gray or the amount of black. We saw this little cartoon example here where there's a synapse connecting X to G. If we assume that the weight of the synapse is 1, then that means that the higher the value of X, the further to the right we are in this image, the higher the value of G, which means the more gray we paint onto that point in space. This random CPPN produces this non-random pattern. Remember that when we're evolving CPPNs, we have a population of CPPNs. They all have different neural architecture, so they all have different connectivity between the input layer and the output layer. The synapses have different weights. So we can imagine two different CPPNs in the population. They have the same input layer and output layer, but depending on the cognitive architecture, the way in which the input layer is wired up to the output layer, the different CPPNs will paint different patterns into whatever space we've defined. Okay. Another detail about CPPNs that will be important for us in a moment is that we can, not only can evolution alter the weights of the synapses, which alters the way that the coordinates are transformed into the output values, but we can also alter the activation functions that sit inside of each of the neurons. In this case, I put a step function inside our output neuron here. Remember, an activation function takes the raw sum that's arriving at a neuron. In this case, it's the value of X times the weight of this synapse. So it's not even a sum, it's just a single multiplication. And we treat on the horizontal axis, we find the value of that weighted sum, and we go up along the vertical axis, and the value on the vertical axis indicates the value that this neuron will output. So the step function here, as you can see, the step activation function means that if the weighted sum is below some threshold, we output a minimum value, which in this case is no gray at all. And above that threshold, we output the maximum amount of that value, which in this case, the maximum amount of gray is this. So we can see how altering the connectivity alters the patterns that we produce, and altering the activation functions inside these neurons also alters the patterns that the CPPNs produce. When we're evolving populations of CPPNs, mutations can alter connectivity and or the activation functions inside these neurons. Hopefully this is ringing a few bells for most of you. Okay. If we start to add in hidden neurons and we add in additional synapses, you can start to produce, or you can start to evolve more complex patterns. Here is a pattern produced by a more complex CPPN. Remember that CPPNs, even if we create random CPPNs, they tend to produce non-random patterns like gradients. If we include the right kinds of activation functions, we can get symmetry. In this case, you can see there's bilateral symmetry. The left side of this image looks like the right side of this image. But there's also some radial symmetry going on here and some other interesting patterns. You might also remember that we can apply CPPNs to spaces of arbitrary dimension. Here's an example where we've added a z-coordinate, which means now we can paint gray within a three-dimensional space. Okay, CPPNs were introduced quite a while ago now, and they're sort of a random tool, and they're a very popular tool in the evolutionary computation toolbox. A couple of years later, in 2014, Cheney, McCurdy, Clune, and Lipson created a different simulator called Voxcad. And my lab maintains a version of Voxcraft that we call, version of Voxcad that we call Voxcraft. And we maintain it at, let me see if I can get this URL right for you. We host it at voxcraft.github.io. As you can see already, the simulator differs quite a bit from the one that you're using. It's a voxel-based, and thus the name Voxcraft, a voxel-based simulator where voxels are volumetric pixels or 3D pixels. Voxcraft, VoxCAD and Voxcraft were designed to simulate soft-bodied robots. So as you're going to see in a moment, each of these voxels is actually deformable. Right? We can increase and decrease the volume of these voxels, twist and pull them in various ways. So Cheney and his colleagues back in 2014 connected the VoxCAD simulator to an evolving population of CPPNs, where the input layer of each of the CPPNs had 3 input neurons, X, Y, and Z, which, remember, means that we're painting a pattern inside an empty 3-dimensional cage. We're now not painting color into this cage, but we are painting or depositing voxels in some of the positions. And then for the voxels that we do place inside this cage, we are colorizing them, and those colors are going to mean something in the simulator. So far, so good. Feel free to stop me at any time and ask questions. Okay, so not too different from what you're doing, right? We're evolving populations of solutions, which are CPPNs. We take each of those CPPN genotypes and we map or transform them into a phenotype, which is a voxel-based soft robot, which is going to be simulated in the VoxCAD simulator. Okay, we have 3 input neurons. Each of the CPPNs has 2 output neurons, one called presence and one called material. The first output neuron down here, whatever value arrives here, we're going to hard code the step function in there. It's not actually shown in this cartoon, but there's a neuron here. And that neuron can either input, that neuron, because it has a step activation function, can only output 0 or 1. 0 means for this, if we put this XY position in here and we obtain a 0 at the first output neuron, that means do not place a voxel there. At a different position, like this position, if we take this XY and Z and feed it in, and we get a 1 at the output neuron, that means place a voxel there. So we're going to move over this space and visit regular points inside this empty cage. And we're going to observe the first of the 2 output neurons. And wherever it's 0, we don't place a voxel. Wherever it's 1, we do place a voxel. If a CPPN does deposit a voxel at a given position, we now have a look at the value in the second output neuron. The second output neuron has a particular activation function that just squashes the raw weighted sum to an integer 0, 1, 2, or 3. So there are 4 possible integers that we can read out of the second output neuron. 0, 1, 2, or 3 means paint the voxel as either green, light blue, red, or dark blue, and there is no particular dark blue in this example robot. So deposit voxels or not, and whenever we do deposit a voxel, paint it with one of 4 possible colors. Okay, so every CPPN in the evolving population has 3 input neurons and 2 output neurons. But between those input and output neurons, each of the CPPN can have a different cognitive architecture. There can be more or less hidden neurons. There can be more or less synapses. There can be recurrent connections. There can be self-connections. So each CPPN is going to be different, and it's going to paint a different pattern inside this empty cage. So far, so good? Okay. So here's an example. Here's an example of one of the soft robots produced by a CPPN. I'll put this on continuous loop here so we can watch it behaving. You'll notice that this particular CPPN placed a voxel at almost every position. I think there's a few hollow concavities in the bottom of this robot. Again, it's mostly made up of light blue, red, and green. What are these colors actually representing? These colors actually represent the material properties of the voxels. This is a particular physical aspect that's missing from PyBullet that you're using. Light blue voxels are meant to be the soft robot equivalent of biological fat. These voxels here, they're very soft and they can be pulled and pushed and they very slowly relax back to their default volume. So like fat in our bodies, they don't actually cause any forces. They're not active. They're passive material, but they provide a little bit of structure. What you can't see in this video, but we'll see in the next video, is dark blue voxels. Dark blue voxels, actually I'll just type this into chat here because I don't think I have this on my slides. Light blue voxels equals passive soft material like fat. The dark blue voxels are passive and stiff. They're meant to be the soft robot equivalent of bone. Passive and stiff. The red and green voxels are meant to be the analog of muscle. So these are active and soft. It's a little bit difficult to see in this picture. You'll see it a little bit in this video. You'll see it a little bit more in the next video. The red voxels, they have a constant sinusoidal pattern that is going into them. And that constant sinusoidal pattern makes them expand in volume and contract in volume together. All of the red voxels increase their volume and decrease their volume in phase with one another. Where does this constant sinusoidal pattern come from? The answer might be a central pattern generator. We've talked quite a bit about CPGs, which we have in our spine, which output a constant oscillatory signal and help us orchestrate walking and other repetitive tasks. In this particular experiment in the Xenobots, there is no neural network controller. The only network is the DNA, the thing that produces the robot's body. We're just going to assume, I guess we could assume that the increase and decrease in volume is being generated by a central pattern generator. But a lot of muscle tissue in mammals will spontaneously increase and decrease in volume when placed together. And this is heart muscle tissue. The particular cell types that make up your heart are a particular cell type that they have kind of their own inbuilt central pattern generator. They are electrically active heart muscle cells, and they will tend to expand and contract together when they're placed together. That detail will come up again when we talk about the Xenobots in a few minutes. Okay, so the red voxels are increasing and decreasing in volume, and the green voxels do exactly the same thing. Green voxels increase and decrease in volume as well, but they do so in antiphase with the red voxels. Yeah. Okay, so we've got light blue is fat tissue, dark blue is bone, and red and green is muscle tissue. Okay, the link here is actually to an older version of the Voxcraft. If you're interested, please check out the GitHub URL that I dropped into chat. Okay, so finally, again, Cheney and his colleagues wrapped an evolutionary algorithm around all this, so they're evolving populations of CPPNs. Here's a video summary of how this project works. Here's an initial random CPPN that produced a random robot at the beginning of evolution. Here's after generation 1, the best robot in the population. Best robot in the population a little bit later. You'll notice these early robots are mostly red and green tissue. There's a little bit of light blue on the top here. I don't know if we'll actually see any usage of the dark blue voxels. So we're looking at the best solution in the population at different points in evolutionary time. Sorry, I should increase this here. Okay. So it should look pretty familiar to what you've seen before, at least in terms of the evolutionary algorithm. So this was an attempt to integrate soft robotic simulation with evolutionary computation to produce evolutionary soft robotics, evolving of soft robots. Okay, this became a building block for the Xenobot project, which we're going to talk about in a moment. Let me highlight one of the co-authors of this paper, Nick Cheney. Nick also took this course several years ago, went off to go do a PhD at Cornell University with Hod Lipson, and has since come back to UVM and is now on the faculty here in the computer science department. Some of you may have taken a machine learning class with Professor Cheney. I think he is teaching evolutionary computation in the fall, but I could be wrong. Okay. In the Xenobot project, which we're going to talk about today, we use basically the same idea. We're going to evolve soft robots in the Voxcraft simulator. But in this case, instead of having 4 possible colors, we're going to have only 2. So we've altered the CPPNs in this case so that the second material output neuron is we're going to add an activation function here that clamps or rounds the raw weighted sum, which is a floating point value, to just 2 integers, 0 and 1. So basically binarizing it. 0 are going to be green voxels, and red are going to 1 is going to paint voxels red. I'll talk about what the green and red represent in this project in a moment. In this project, we used the AFPO evolutionary algorithm, which I mentioned briefly a couple classes ago. So in the center, we have Voxcraft. We are then evolving populations of CPPNs using the AFPO evolutionary algorithm. Here is what the fitness curves look like for AFPO. We have evolutionary time on the horizontal axis here. So we have 1,000 generations. One of the nice things about AFPO is it maintains populations of genetically independent species. If you think about your parallel hill climber, if you have a population size of 10, you have 10 parents and each has a child at any given time. Parent I and parent J never combine genetic material. They're genetically independent. So you can think of a parallel hill climber that has a population size of 10, of having 10 unique species in the population at any given time. AFPO is similar in that it also maintains genetically independent species, but the number of species can increase and decrease over evolutionary time. And the number of individuals that belong to each species can also increase and decrease over time. If a species is doing well, like the pink species that you see here, there may be many members, there may be many pink robots, many members of the pink species here. We won't go into too many more of the details of AFPO, but again, for those of you that are interested in evolutionary computation, AFPO is a good one to look into. Okay, at the end of one run of the evolutionary algorithm, at the end of one run of AFPO, we extract from the population the fastest moving soft robot. So as always, our fitness function is just going to be fast locomotion. Fitness is plotted on the horizontal axis here, and the distance is actually measured in the number of body lengths. So in this particular run of AFPO, at the end of 1,000 generations, there was one soft robot in the population that could travel 7 body lengths in however many time steps we had run the simulation for. I don't remember the exact number, but it's not important for our purposes. Okay. This is it. This is what it actually looks like. This is just to remind us of the different body parts, the different kinds of voxels that can make up this robot. You'll notice, I don't know if I can zoom in here. Can I zoom in? Okay, no, sorry, you're going to have to squint here. You'll notice that this robot is made up of all red voxels. What are red voxels? In this case, as I mentioned, in the Xenobot project, we're going to take some of these best evolved, fast-moving simulated robots and build them from frog cells. In this case, in this project, we were able to use 2 different types of frog cell types. The first one are frog skin cells, which are going to be the green ones. Kind of a coincidence that the frog skin, we labeled it with green. The frog skin cells, again, that skin tissue we're going to assume is elastic. It's not completely soft. It's like our skin, but it is deformable and passive. It doesn't apply any forces in and of its own right. The second cell type we're going to use are what are known as myocardiocytes, which is a fancy term for heart muscle tissue, heart muscle cells. So that's the red here. This is going to be the active component of our robot. So like the previous project, we're going to assume we told AFPO, we told the evolutionary algorithm that it can build soft robots out of these 2 parts. Since the fitness function was only locomotion, most of the solutions that evolved were just balls of muscle. So you can see this evolved design here is all red. The AFPO decided not to use any skin cells whatsoever. It made a ball of myocardiocytes. If we go back and watch the video for a moment, you'll notice that in this older project, some of the initial robots had some passive soft material, but after just a few generations, evolution had discovered that if the fitness function is just locomotion, if there's no penalty for energy usage, you might as well design robots that are basically just balls of muscle. That was discovered by this evolutionary algorithm in 2014 and was rediscovered by our modified algorithm in 2020. Sam, who was evolving the soft robots in this case, went back and performed a second run of the AFPO algorithm that produced a second robot. He ran AFPO a third time. It produced a third robot, and so on. And he ran this algorithm 100 times, as you can see here. Each of those 100 runs of AFPO produced 100 champions. And in every single case, AFPO discovered that the best solution is to ignore the green tissue altogether and just make robots from red voxels. You'll notice that the shapes are slightly different. You'll notice, I think actually if I exit out of here, I should be able to zoom in. There we go. You'll notice for each of these designs, there's a little rainbow trajectory. This is the way in which the robot moved in the simulation. So we were trying to plot form, the 3D shape of the robot, and also function, the way in which it moved. Okay. So once we had these 100 designs, we talked to our biology colleagues, Mike Levin and Doug Blackiston. Doug, who's the microsurgeon, who's the one who actually builds Xenobots, he looked at all 10 of these and shook his head and said, "There's no way that I can build these designs. I can't build up physical Xenobots just from myocardiocytes. We need to use combinations of skin and heart muscle." And I'm going to show you some videos of Doug actually building Xenobots in a few minutes, and you'll see why he can't build from just myocardiocytes. But for our purposes for the moment, let's assume that we failed to cross the sim to real gap. Not only did what we build fail to do what it did in simulation, we couldn't even build them in the first place. So we completely failed to cross the sim to real gap. We didn't understand why, so we sat down and had some involved technical discussions with Doug, and he explained more about how he actually builds the Xenobots. Sam took what Doug told him and built it back into the simulation and altered how red and green voxels are simulated in Voxcraft. So this is an example of real to sim. We learned from our biology colleagues what is and isn't possible, what's buildable and what's not, and we built that back into the simulator, and then we tried a second sim to real attempt. Okay. Okay, so what details did Sam build into his simulator? The first thing that Doug told us is that the way we were simulating the expansion and contraction of these voxels was way too strong. These robots were bouncing and jumping off the ground, like you saw in the previous video. He said heart muscle cells don't work like that. They're not that strong. So we tuned down the strength of the voxels by a huge amount. The other detail that we got wrong in our simulation was that we assumed that all of the red voxels would increase and decrease in volume in phase with one another. That is true if myocardiocytes arrange themselves into a frog heart for frogs. They recognize somehow, and we're not sure how yet, but they recognize that they're in the right configuration and they synchronize, and that's good news for the frog because a heart that increases and decreases in volume acts as a pump. When all of the myocardiocytes, which make up the shell or the perimeter of the heart, when all of those cells contract in volume, they pull together and pump blood out of the heart. As they relax and increase in volume again, then the heart expands in volume and pulls blood into the heart. That is true for humans as well. Unfortunately for humans, there is a common pathology among older adults known as atrial fibrillation, in which that synchronization breaks down and different parts of the heart start to increase and decrease in volume out of phase with one another. When that happens, as you can imagine, the overall volume of the inside of the heart doesn't change that much. If you have a skin of cells and they're pulling and pushing in random phase offsets to one another, the total volume of the heart doesn't change, which means blood does not get pumped out and blood does not get pulled in. This is a common problem. Especially that happens in normal hearts, but if you take myocardiocytes and you rearrange them into new shapes, like this shape, myocardiocytes have never been arranged in this pattern before. The cells will still increase and decrease in volume, but they won't sync up anymore. So the second important detail we built into the simulation is whenever a CPPN placed 2 or more red voxels into a simulated Xenobot, we would set random phase offsets for each of the cells. So each of the 2 red, if we just had 2 red voxels, both would increase and decrease at the same frequency. But we would set some random phase offset. I can't do this too well except for antiphase. They'll increase and decrease at random phase offsets. So now the red voxels collectively are weaker and less coordinated than they were when we did our first sim to real attempt. So far, so good? Just pause for a moment in case there's any questions. Feel free to type a question into chat or unmute yourself if you like. No? Okay. So now Sam was armed with a more accurate simulator. He did another 100 runs of AFPO, and in this case, it produced these 100 champions, the best one from each of those 100 runs. And you'll see now that evolution is incorporating this passive material. So there is something about this passive material that helps produce forward locomotion when the individual components, the individual red voxels, are not as reliable as they were before. So let's have a look at one of these second AFPO runs. Here's a random CPPN. You can see it's painted mostly, I'm sorry, they're not green in this case, they're cyan or light blue. The light blue voxels are the frog skin cells in this case. And you'll notice the red voxels here. In this example here, Sam was painting them red and green. Red indicates that they're expanding in volume, and green indicates that they're decreasing in volume. So this is a random CPPN, a random robot. It is not traveling very far from the origin at all, so this one gets a very low fitness score. Here's a second random design. You'll notice there's more motion in this case, and it actually does displace a little bit away from the origin. So it gets a small non-zero fitness value in this case. Here's another example of a third random robot, which is a ball of muscle, and you can see that a ball of muscle is no longer a good solution. This particular robot, although there's a lot of self-motion, there's no self-displacement, not much movement away from the origin. Okay. Sorry. So those were three random solutions, which evolved eventually into this robot. In the second AFPO run, we got this robot, third, fourth, fifth. So now we had a second set of 100 candidates for Doug to build from frog cells. Turns out that again, in this second attempt, the vast majority of these, Doug shook his head. He said he couldn't build them. So instead of just having a conversation to figure out whether we cross the sim to real gap, Sam on the computational side and Doug on the biology side, they sat down and they made a flowchart. The idea here is that you take each evolved design and you compare it against the flowchart, and you ask at each stage in the flowchart, you ask the various questions. So the first question is, does that design have contiguous tissue regions? Meaning Doug could not build a checkerboard pattern. If the let's find one that looks very checkerboardy. This one, these are not, most of these are pretty good. Actually, here's a good example here. You'll notice there's some small regions of light blue surrounded by regions of red. So if a design has blue, red, blue, red, blue, red, blue, red. I'm sorry, I know we switched from red and green, green and red to blue and red. Blue are the skin cells in this case. Blue, red, blue, red, blue, red, blue, red, where there were very few contiguous regions of red or blue, that was extremely difficult for Doug to build. He's going to build these up from cells. He can't put one skin cell right next to one muscle, skin, muscle, skin, muscle. He doesn't have that high resolution manufacturing ability yet. So we had to build these very low resolution, big patches of muscle or skin. So if there were not these big patterns, if there were not big contiguous regions, the answer is no, and that design is knocked out of the competition to be built. If the answer is yes, a design has contiguous regions, the next question to ask is does that design have a stable geometry? Or meaning, will it have a stable geometry when it's actually built from frog cells? What is a stable geometry? A stable geometry means that if there are any gaps in the design, so here's a gap in here, there's a hollow core, here's a hollow core in here, here's another gap or a concavity, anything that bends into the center of the body, any concavities, they have to be 100 microns or larger, which is about a tenth the size of the Xenobot. Xenobots are about 1 millimeter across. I'm sorry, I think I forgot to mention this at the beginning. So they're about the size of a grain of sand. Why can't we have very small concavities inside, or very small gaps inside inside the Xenobot? The reason, and you're going to see this actually in a video in a minute or two, is that frog cells and cells in general, they don't like to be on their own. If they have only a few neighbors, they will try and pull inward and connect with other neighbors. Generally speaking, most biological tissue in you is contiguous. If you had tissue that had lots of holes in it and individual cells drifting apart, you would literally sort of fall apart. It's not something that cells like. They like to clump together and make stuff. So if you have a small concavity, the cells will gradually readhere and close off the cavity, and you'll fail to cross the sim to real gap because it won't hold that small concavity. But if there's a big concavity like this, that's okay. That's a big enough distance that the cells can't figure out how to close up again. Okay, so if there are big splotches of skin and muscle and there are no small concavities or no small holes, you can move on to the third question, which is mostly passive tissue. In this experiment, we wanted to build Xenobots that had at least 50% passive tissue, which is the skin cell. So you got to be at least 50% skin for energy efficiency. We didn't want to build balls of muscle. And this would also allow us in future experiments to scale up to more complex tasks because we could store stuff where that passive tissue is. At the moment, Xenobots have no sense organs, they have no digestive system, they have no nervous system. They're almost like a wind-up toy. So the skin, the light blue patches, are sort of holding spaces for stuff we could put in later. Okay. If the answer to that is yes, if a given design passes all three of these stages, then at least as far as Doug could tell, it can actually be built. Among the 100 designs, and in this table here, we ranked those designs by how fast they move. So it turns out that robot number 90, which is 1, 11, 21, so 90 is this guy over here. This was the fastest mover, but was not buildable. It invalidated condition B1 and B3 as well. The second fastest mover met this condition, but failed condition B3. We continued down this list, and it turns out that the 15th fastest moving Xenobot, which is design number 52, passed all three of these tests, which is this robot down here. For whatever reason, this particular robot moves quickly in the simulator and, according to Doug, was buildable. Okay, here's how design number 52 actually moves. You'll notice it has most of the myocardiocytes on the ventral or bottom surface. They're increasing and decreasing in volume in random offsets to one another. But because of the 3D shape of this robot, it tips forward. It's got these two big legs on the back, and the passive tissue kind of dampens out the random action of the heart muscle tissue underneath. And for some reason, the combination of shape and distribution of these two cell types leads to consistent motion from left to right. As I mentioned several times throughout this course, One of the reasons why we want to use an evolutionary algorithm for designing robots is that designing robots is a very non-intuitive problem. This is about as non-intuitive as you can get. We are trying to build a machine from parts where some of those parts are unreliable. We don't actually know when we build this physical Xenobot how these heart muscle cells will coordinate, if at all. So we just simulate them as no cooperation whatsoever. It's sort of an overly conservative guess about how this part of the robot is going to behave. Another way to think about this from a design perspective is imagine you were trying to design a boat, and you could put into this boat human rowers, and all of these human rowers they're going to row at the same frequency. You can put as many rowers into the boat as you want at any position in the boat, but these rowers will not synchronize their rowing. They're going to row at different phase offsets from one another. What is the shape of the boat, and where should you put human rowers so that the boat moves forward and it moves forward quickly? That's sort of the analog of the design problem that AFPO is trying to solve in this case. Somehow it's able to solve it. Okay, that's the computational side. So now we've got our design. We're ready for Doug to start building a physical Xenobot. So I'm going to dip into some of the biology now, but before I do, I'll just pause again for a moment if there's any questions about any of that. So far, so good? Okay, so what I'm going to show you is a series of 10 short videos that are taken during the construction of actually the first Xenobot, which is this one over here. The first attempt to build one was not actually to try and build this one. Hannah says this reminds her of emergent behaviors in complex systems. Absolutely, this is an example of emergent behavior. The emergent behavior is consistent travel, consistent locomotion that arises from randomly acting or random components. Absolutely, this is a complex system. Yep. Okay, so way back at the beginning of this project, about 2 years ago now, when the two research teams were first getting together, we had weekly Skype calls where we were explaining what physical simulation is, what evolutionary robotics was. They were explaining what they're able to do with frog tissue. On one of the very first Skype calls, we showed one of our virtual creatures running around in simulation. Doug went and built what he saw. We showed him one of our little four-legged hopping quadrupedal robots. Doug went to see if he could actually build this out of frog cells to begin with before we got to this stage. In this case, he's going to build just with skin cells. So this is not actually a robot. It's a sculpture. This thing doesn't actually move. But I'm going to show you how Doug built this robot to give you a sense of how this works. He starts by harvesting early embryos from Xenopus laevis. Xenopus laevis and other frogs lay millions of eggs. He took some of those eggs, or took a couple of them, very early embryo. So as you can see, these are very, very early on. There's only a few thousand cells at this point, way, way before they become an egg and a tadpole. So this is early embryo. You'll see a bunch of these embryos in the video in a moment, and you're going to see Doug going from one embryo to the next and injecting a particular chemical signal that determines cell type. It triggers particular cells to develop into particular cell types, and the cell type that Doug was interested in this case is frog skin cells. Hopefully I can play these videos. There we go. Okay, so here is Doug injecting these chemical signals. So he's biasing the cells around where he injected to develop into skin cells. He then is looking through a microscope, and each one of these embryos is about 1 millimeter across. You'll notice that he's reaching in with micro forceps. So Doug is one of the most skilled microsurgeons on the planet. You have to have extremely steady hands to do this work. You'll notice he is gradually pulling off this vitaline membrane. It's sort of the sac that keeps all of the cells in the early embryo together. Once he's pulled off the vitaline membrane, he's going to go in and cut off a cap or the tops of these embryos. I'm sorry, I don't know why I'm having Wi-Fi issues this morning. Please bear with me. Okay. So he's going to pull off what's known as the animal cap. It's the part of the embryo that actually would have developed into the animal in this case. Again, each one of these embryos is about 1 millimeter across. It's extremely delicate work. Okay, so there's the separation of one animal cap. He did this for several of these embryos. Next step, stage 4, is to separate the ectoderm. This is the dark material that you can see, part of the skin layer of the embryo. And he's separating out all of the inner cells, the cells just underneath that ectoderm layer, which you can actually see in white. And the reason why he's trying to get at those cells, as you can see, is they're less sticky at this point. You can actually see in this video some individual white dots. Those are individual cells. These are, at the moment, these are stem cells, but they are fated. They're going to become skin cells because they have been treated by Doug's injection. So these white cells, these stem cells, are going to be the raw material that he's going to build the Xenobot from. He then collects up all of these cells into a syringe. These dissociated cells, he's now going to inject these cells into a very small concave depression, which is the dark region that you see here. They're all going to settle into that concave region. This is almost like putting a muffin mix into a muffin container. It's all going to congeal into this mold. I think this is another video of more or less the same process. Yeah, you can see multiple wells here. I'll just speed this up a little bit. You can see the individual stem cells. And you'll notice in this, you can see it better in this video, they're actually starting to congeal back into the cell. Over a period of hours and a couple of days, you'll notice that these cells do what they want to do, which, as I mentioned a few minutes before, is they do not like to be on their own. And they will try and re-adhere. They will try and combine back together again. My biology colleagues refer to this as rebooting multicellularity. So we've got all these individual cells and they want to come back together into a multicellular mass. What do these re-adhered cells want to do? This is $1 million question in biology. Okay, somewhat separate from what we're going to look at for the rest of today. Okay, as they're left for a couple of days, they form into a mass of about 2,000 cells, which you can see here, and they start to compactify. They start to pull inward into a tighter and tighter, more solid mass. This is the raw material now that Doug is going to use to sculpt shape. We have a pure strain of stem cells that are developing into skin cells. Once they've matured and become dense and attached together enough so that they're a solid mass, now Doug can go in and start to do some sculpting. And you'll notice in a moment he's going to reach in with two tools. We've got the micro forceps on the left and a micro cauterization device on the right. The cauterization device is simply a hot wire that is burning away some of the material. So we've got basically a sphere of spheres, and Doug is going to sculpt by subtraction. He's going to remove excess material. You'll notice he's making a crosswise pattern. This is the crosswise pattern on the ventral or the undersurface. So he's making these four stubby legs by burning away the excess material between where the legs should be. Okay, I think you get the idea here. Okay, and you see now he's doing a little bit more sculpting. And then finally some finishing touches on this to make the sculpture itself. Oops, sorry. Logan asks, what is the fluid this is occurring in? It's occurring in the fluid that is most conducive to frogs, that frogs like best, which is room temperature fresh pond water. Well, it's not actually pond water, it's just room temperature fresh water. So this is simultaneously a frog and not a frog. Yeah? Okay. All right, so I just walked you through Doug creating a sculpture of one of our four-legged robots. It can't move because it's just skin cells. So let me just back up a little bit to this slide. Remember that he needs to build this particular design here, which has skin on the top and muscle on the bottom. You remember that partway through this process, I showed you him injecting cells from a syringe into a concavity. The reason that what he can do is he can actually separate out skin cells and separate out a separate collection of myocardiocytes or heart muscle cells. And then he can layer down different layers of heart muscle, skin, heart muscle, heart muscle, heart muscle, skin, skin, heart muscle by layering down from the skin syringe or the heart muscle syringe. So basically building it up like a sandwich. That's the reason that he needs contiguous regions. He cannot place one skin cell and one heart muscle cell, at least not yet. Okay. By doing that, by altering the process that I just showed you, by incorporating muscle cells into his toolkit, he can build not just a sculpture, but something that actually moves. Okay, so I'm going to play this video here, which is going to re-show you what the simulation predicts the evolved Xenobot will do and what the actual Xenobot does, which is more or less what the simulator predicted it would do. So in this second round of sim to real, we managed to cross the reality gap. I'll play that for you again. You'll notice that obviously the Xenobot's shape is not quite like its simulated counterpart, and it doesn't quite move in about the same way. We're actually looking more from above in this case than we are. We're looking more from the side in simulation and more from above in reality, but it's close enough to get consistent forward travel. So this is a millimeter-sized Xenobot that is walking along the bottom of its petri dish. Any questions about that? Okay. So in the paper, we included this video, but we also included some results to show that not only could we cross the reality gap once, but we could cross the reality gap multiple times. So how did we do that? We took our simulated Xenobot and we reran it multiple times in the simulation with different random action of the heart muscle tissue, which meant that the simulated Xenobot moved the same simulated Xenobot, but with different random action, moved in different ways, which are the pink trajectories. So in panel C here, we're looking directly from above. We put the simulated Xenobot at the origin 0, 0 multiple times, and it tends to walk to the right, but depending on how the simulated heart muscle tissue works, sometimes it curves counterclockwise, sometimes it curves clockwise. So these are all the pink trajectories. Doug then took the built Xenobot, also placed it at the center of the Petri dish, looked down through the microscope, and saw that over multiple trials with this Xenobot, it tended to also walk to the right. Not always in perfect curves like the simulated Xenobot. So there's more or less a match between the pink simulated trajectories and the blue physical trajectories. We could argue about how close this match is. Maybe you would argue that it's not very close. So we performed a control experiment where we flipped the simulated Xenobot on its back. And now it doesn't move very much at all. It sort of jiggles in place when it's placed on its back like a turtle turned upside down, which produces the pink trajectories that you see here, much shorter and no longer biased to the right. Doug took his physical Xenobots, also flipped them on their backs, and they also did not move. So what does this particular control tell us? It tells us that the 3D shape and the distribution of heart muscle and skin matters. Because although this is the same Xenobot, if we flip it on its back, from the point of view of behavior, it now has a different shape and a different distribution in space of red and green. And by disrupting the form and distribution of the evolved Xenobot, we disrupted function. Okay, so that was our proof that we managed to cross the reality gap. We've got 4 minutes left, so I'll show you just a couple more videos of some of the other things we tried now that we were able to evolve Xenobots and cross the reality gap. We took some of our evolved designs, and Doug made multiple copies of them. So he built multiple instances of the same Xenobot design and then put those clones in a group. Remember our discussion about the virtual lions and the virtual gazelles on the virtual Serengeti plain? He put them together, and we also sprinkled some very small particulate matter. These are they're hard to see in the video here, but you'll notice there's some very small pieces in the dish. These are small glass beads, which are much smaller than the Xenobots. You'll notice, first of all, that given the 3D shape of the Xenobots, just because of their shape, they sometimes, when they crash into each other, they temporarily lock together and then move apart again. This is an idea, this triggered an idea for us in that, again, how the body shapes the way we think, or how the body shapes the way that these things might act together. Here's a better picture of what happens with this swarm of Xenobots. Here you can see the red particulate matter. Through the collective action of the swarm, they've managed to push these particles together into piles. Who knows what the applications for Xenobots may be in the future? We mentioned a few ideas, but it's hard to say what the eventual useful applications will be for brand new technology. We mentioned that maybe in future Xenobots might be used to search for and remove contaminants from the soil. Since these things can exist in room temperature fresh water, we could introduce them into polluted waterways and have them find and collect microplastics and pull that out of waterways. So there may be some environmental applications. It may become possible in future to build biobots out of not frog cells but human cells. If we can, those human biobots might be able to be introducible to the human body. So we looked at some. Oh, sorry, I'll show you one more video here. Here's some more of this particulate matter. You'll notice the Xenobot in the bottom seems to be acting like a sheepdog. If we can make biobots out of human cells, we might be able to tackle the problem of intelligent drug delivery. Imagine we make a biobot out of the cells of a human patient. Imagine we make a hollow biobot and we put a small piece of medication represented by the yellow voxels in this case. If the human patient were then to swallow a biobot made from their own cells, and that biobot can move through the patient's body and deliver that drug to wherever it needs to go in the patient's body, we can then achieve intelligent drug delivery, get the drug to where it needs to go. And we can avoid triggering the human patient's immune response because the introduced foreign body is not foreign. It's made from the human patient's own cells. Again, that application is years away, but it's interesting to think about. We've reached 9:45, so this is, I think, a good place as any to stop. You have a quiz due tonight, your final quiz. This Sunday night before midnight, you are submitting your written report and video, and then I will see you all back here next Monday morning, very bright and early. I've spent a lot of time talking in this course. I look forward to seeing what you have to present. Sid is asking if there will be office hours on Thursday. There will indeed be office hours on Thursday. I recommend if you have any questions or final questions about your final project, please do come and see me then. Otherwise, I wish you a good rest of your week. I wish you a good end to the semester. Good luck with your other exams. Good luck with finishing up your final project. See you all next Monday. Thanks very much, everyone. Bye-bye.